day everyone, we are the group 3 in biochemistry and we will be explaining microbiology, the types of microscopes and their function, basic lab apparatuses, and microbiology instruments. So what is microbiology? Derived from the Greek word mikros, meaning small, and bios, meaning life, and logia, or logos, meaning study of. Microorganisms are minuscule organisms that cannot be seen with the naked eye. An optical instrument that has the capacity to magnify objects up to 1,000 or even 100 times. The study of organisms led to the discovery of the microscope. The microscope has seen significant revolutionary changes since its original discovery in the 1600s, becoming more sophisticated and involved with time. The basic prototype created by Van Leeuwenhoek gave rise to a variety of microscope variants. There are 9 types of microscope. Starting with compound microscope. It contains a magnifying lens. It can magnify objects approximately 1,000 times their original size. Visible light is its main source of illumination. On to bright field microscope. This is used to visualize bacteria and fungi. Made up of a series of lenses, and it can magnify an object 1,000 to 1,500 times. Dark field microscope. This type of microscope is used to view spirochete. It utilizes reflected light instead of transmitted light. The specimen to be studied appears bright against a dark background. Contrast microscope. It has a contrast enhancing optical technique in order to produce high contrast image of specimens that are transparent, which include thin tissues, slices, living cells in culture, and subcellular particles. Differential interference contrast microscope. It's similar to phase contrast microscope, except that it utilizes 2 beams of lights and has a higher resolution. It is used for examining living specimen when normal biological processes might be inhibited by standard staining. Fluorescence microscope uses ultraviolet light and fluorescent dyes called fluorochromes. It can be used to visualize structural components of small specimens, such as cells, and to detect the viability of cell population. It may also be used to visualize the genetic material of the cell. To confocal microscope, known as confocal laser scanning microscope or CLSM, or laser confocal scanning microscope LCSM, uses an optical imaging technique that increases optical resolution and contrast of the micrograph by using a spatial pinhole to block out of focus light in the image formation. Electron microscope. It utilizes a beam of electrons to create an image of the specimen. The electron beams serve as the source of illumination, and magnets are used to focus the beam. Modern electron microscopes are capable of magnifying objects up to 2 million times. Scanning probe microscope. It is used to study the molecular and atomic shapes of organisms on a nanoscale. A physical probe is used to scan back and forth over the surface of a sample. It can also be used to determine the variations of temperature inside the cell, as well as its chemical properties. To create samples to study microorganisms, we need agar, a gelatinous substance made primarily from agarose and agaropectin, which are obtained from cell walls of red algae belonging to the Gelidiaceae and Racilariaceae families, which is suitable for performing molecular tests and cultivating microorganisms. Specimens refers to a part or a body of a living or non-living organism that undergoes a rigorous study that serves as a point of reference to a various aspect of biology. Distilled water. It is produced by heating the water into a boiling point, letting the steam ascend, which allows the steam to rise, then recondense into liquid in a cool down chamber. This water eliminates the impurities that could affect the precision of the findings.
So now let's move on to the basic laboratory apparatuses. The first one, we have the beaker, which is used mostly in chemical, biological, clinical, and industrial laboratories, as they are function as a container to store or mix liquids. We also have the burette, which is a glass tube marked with measurements, functions as a tool for accurately measuring the volume of standard solutions, and is commonly used in the titration process for quantitative analysis. A glass panel that is used to funnel liquids or powders into narrow opening containers in chemistry and in other scientific experiments. We also have the separatory panel that is sometimes referred to as a sep panel, separating panel, or a separation panel. It is a piece of laboratory equipment used in liquid-liquid extractions to partition or separate the ingredients of a combination into immiscible solvent bases with varying densities. Graduated cylinder, an equipment used in laboratories to measure liquid or solid volumes through displacement. Pipette, which is a piece of equipment used in laboratories to measure and transfer small amounts of liquid in milliliter or microliter proportion. Steering rod, which is used in laboratories when combining liquids or solid and liquids test tubes, which are used to mix chemicals, hold, heat liquids, grow bacteria, and collect or store bodily fluids like blood and semen. Thermometer that is used to measure both the boiling and freezing point during laboratory experiments. It is also used to reach specific temperatures needed for certain reactions to occur. A round concave glass used for weighing solids and as a lid for flasks and beakers that is called watch glass. It is also used to initiate the process of evaporation. Have the wash bottle, which is used to rinse laboratory equipments and glasswares. It often appears in the form of a squeeze bottle with a nozzle. Crucible and cover, on the other hand, are usually made from materials like porcelain or metal. However, crucibles allow chemicals to be heated to very specific temperatures, but the cover is usually set loose in order for gases to escape during heating. The evaporating dish or dishes are pieces of heat-resistant glassware that are commonly used to hold substances that are being evaporated. Spot plate are ceramic equipments with small depressions that are used to perform controlled tests to substances by using only a small amount. A Bunsen burner is a classification of gas burnt used primarily to heat and combust substances. In addition, they are also used to sterilize objects and are usually fueled by methane, butane, propane, or a combination of gases. The burette clamp is a laboratory tool used to hold the burette peaks during laboratory procedures involving the volume measurement. It is usually made from plastic or metal and can be tightened as needed. The clay triangle, or also known as pipe stem triangle, is a triangle made from wire and ceramic that is used to support a crucible while it is being heated over a Bunsen burner. The crucible tongs are steel tools used to lift hot crucibles from a burner or furnace to prevent injury to the hands. The lab tongs, on the other hand, are also called the pincers, are mostly used in lifting and grasping in high temperature reactions. The beaker tongs are also called the beaker clamps. As its name suggests, a beaker tongue is used to move, transfer, and hold beakers containing hot substances. Deflagration spoon, or a combustion spoon, is a brass spoon used in chemical experiments where highly combustible materials like phosphorus and sulfur are involved. The iron ring, which is a laboratory apparatus commonly used for supporting other apparatus such as a funnel or filter.
Iron stand, also known as a retort stand. An iron stand is a metal laboratory equipment used to hold other equipment such as burettes, test tubes, flasks, and other types of glassware. Platform balance. A platform balance, or laboratory balance, is a form of equal arm balance that is used for weighing objects. Spatula. A spatula is a crucial apparatus that is used in a variety of ways. They are commonly used for mixing, stirring liquids, lifting, handling, or leveling granular materials. Test tube brush, also referred to as a spout brush. It is used for cleaning narrow mouth glassware, like test tubes, cylinders, burettes, and flasks. Tripod. Tripods are used to provide a stable surface for experiments. They are often used in tandem with other equipment like Bunsen burners, hot plates, and wires. Test tube holders are used to hold test tubes when they are too hot or cold to be touched. A water bath is a piece of laboratory equipment made from filling an enclosed container with water. Aspirator, also known as an editor jet pump or filter pump, is a device capable of producing a vacuum. It is usually used to speed up the flow of a liquid through the Venturi effect. A narrow tube is located inside that contains the liquid. Mortar and pestle, like in the kitchen, is used in a laboratory to grind up and pulverize solid elements into fine powder. Test tube racks. Racks are laboratory equipment used to hold multiple test tubes at the same time. Microbiology instruments. Analytical balance. An analytical balance is a highly precise measuring instrument used to determine the mass of small samples with a high degree of accuracy and precision. Autoclave is a machine that uses steam under pressure to kill harmful bacteria, viruses, fungi, and spores. Centrifuge. The centrifuge is a commonly used tool in laboratory research. It uses centrifugal force to separate substances in liquid or solid nebula according to particle size and density differences. Colony counter. Count the number of colonies of microorganisms that have grown on an agar plate. Laboratory deep freezers play important roles in safe storage of reagents, frozen vaccines, and other temperature-sensitive specimens. Hot plate is a laboratory device used to heat samples, solutions, and materials uniformly without the danger associated with the open flame at precise temperatures. Hot air oven utilizes dry heat to sterilize laboratory equipment, glasswares, and samples, making it essential for procedures that cannot tolerate moisture. Incubator. An incubator is a heated and insulated container that controls the temperature and the humidity when working with cells and microbiological cultures. Laminar airflow. A laminar airflow, or laminar hood, is an enclosed station that filters and captures all particles entering the designated space to create a contamination-free work environment.